Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Ladies and gentlemen, good morning. A very warm greeting to each and every one of you. Welcome to the first webinar series of the Research Center for Pharmaceutical Ingredients and Traditional Medicine, National Research and Innovation Agency, the Republic of Indonesia. My name is Yudi. It is a pleasure to be your host today. On behalf of the organizing committee, we extend our appreciation for your presence today. On this special day, we have three speakers who are experts in their fields about drug discovery and Auto-Affinity labeling technology. First of all, let me give a very warm welcome to the speakers who are already with us today. To Professor Dr. Koichi Watashi from National Institute of Infectious Diseases, Japan, to Professor Dr. Mohammed Hanafi from Medicinal Chemistry and Artificial Intelligence Research Center for Pharmaceutical Ingredients and Traditional Medicine, Indonesia, and to Professor Dr. Makoto Hashimoto from Hokkaido University, Japan. They will be sharing their experience with us, which is a great opportunity to learn these important topics from the experts directly. Without further ado, let us start our program by welcoming the Head of Research Center for Pharmaceutical Ingredients and Traditional Medicine, Professor Yuli Widiastuti, to deliver her opening remarks. Please welcome Professor Yuli. Thank you.
Thank you, Mas Yudi, for this opportunity. Honorable guest speaker, Professor Koichi Watashi, Professor Muhammad Hanafi, and Professor Dr. Makoto Hashimoto. Distinguished Professor, dear colleagues and participants, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh, and good morning, everyone. Indeed, it is my great honor to welcome all of you to our today's webinar series of research talk with the topic of drug discovery and development. On behalf of Research Center for Pharmaceutical Ingredient and Traditional Medicine, I would like to extend my sincere gratitude to Professor Koichi Watashi from National Institute of Infectious Disease and Antiviral Drug Discovery Research, Professor Dr. Makoto Hashimoto from Hokkaido University, and also Prof. Muhammad Hanafi from Research Center for Pharmaceutical Ingredient and Traditional Medicine as our guest speaker in this webinar. Kindly inform that this is the fourth research talk which is held by Research Center for Pharmaceutical Ingredient and Traditional Medicine. Today's talk is organized by Medicinal Chemistry and Artificial Intelligence Research Group. Still the same as the previous webinars, today's talk is take a theme related to the exploring and developing natural product chemistry for drug design and development. Ladies and gentlemen, up to present time, Indonesia has relied on almost 95% of its drug raw material need on import from various countries, especially from China and India. On the other side, Indonesia is well known as a mega biodiversity country, included the medicinal plant and marine biodiversity. Moreover, due to the support the new policies in health transformation, especially the pillars of health resiliency, one of the efforts that can be done is to encourage research on the development of drug raw material. We all know that in the past 2 decades, drug discovery effort has spent millions of dollars through high throughput screening that resulted in only few patented drugs. Currently, the technological advances in the field of drug development research, it was allowing artificial selection of molecules with clear protein target, so it will cut the cost enormously. However, the discovery of new novel compound is still needed. To anticipate new emerging diseases, including re-emerging diseases, which of course require new therapy and new drug. We believe that nature is the source of all medicinal chemical compound, and Indonesia, with its biodiversity, is the largest chemical factory in the world. Ladies and gentlemen, I'm pretty sure that this webinar will give us new perspective about the research experiences from Sensei Koichi Watashi, Professor Muhammad Hanafi, and also from Sensei Makoto Hashimoto from their respective field. I sincerely hope that this research talk will provide a good opportunity for researcher, scholar, and graduate in natural product chemistry to get and also to share a valuable experiences, knowledge, and views on drug discovery and development based on synthetic chemistry and also from exploration of natural materials. Ladies and gentlemen, last but not least, special appreciation goes to committee, especially to Dr. Teni Ernawati and her college who could organize this successful event. And please, now allow me to open this webinar officially. Hope you all to have an enjoyable webinar. Thank you. Wassalamualaikum warahmatullahi wabarakatuh. Arigatou gozaimasu. Time back to Dr. Yudi. Thank you.
Thank you very much to Professor Yuli for the opening remarks. Before we move to the next agenda, I would like to invite and appreciate you to turn on your front camera for a photo session. I will do the screenshot through my laptop screen. So we have 4 pages here, and I will start from the first one. So please, the audience, to turn on your camera. Okay, and I will start from the first page. Okay, the second page. The third page. And the last page. Okay, fantastic. Thank you. Next, we are going to the main session. And before we start, let me introduce our moderator today, Dr. Zetriana Putri Tahmin. Dr. Zetri will run the next session of scientific speech from our speakers today. And please welcome Dr. Zetri. Thank you.
Thank you very much, Dr. Yudi, for opening this great today's seminar. Good morning, everyone. Before we begin, I would like to inform you that we will have 3 speakers for today, and there are 2 sessions for each speaker. The first session, each of the speaker will deliver their speech, and then followed by question and answers session. You can write down the question in the chat box by mentioning your name, institution, and then your question. I hope during the speech, all the attendants can turn off the Zoom mic by ensuring the mic icon was crossed in your screen. Then, further ado, I would introduce you to the first speaker, Professor Koichi Watashi. Today, Professor Koichi Watashi will deliver the speech about the drug discovery and development of antiviral. First, I would like to introduce him to all the attendants here. Professor Koichi Watashi is the Division Director in Division of Drug Development, Research Center for Drug and Vaccine Development, National Institute of Infectious Disease in Japan. He also has a dual appointment as a visiting professor in Tokyo University and Waseda University, and also adjunct faculty member in Nagoya University. He won several awards in 2016 in Taisho Toyama Award, and also in 2017 in MSD Award, the Japanese Society of Hepatology. In the screen, you also can see the recent publication of his work in 2022. Without further ado, I'd like to welcome our presenter to start the presentation. Professor Watashi, are you here in the Zoom?
Yes. Can you hear me?
Yes, I can hear you very clearly.
Okay, so I will share my slide. Is it okay?
Yes, I can see your presentation also very clear.
Yeah.
Professor Watashi, please start the presentation.
Yeah, thank you very much. So at first, I appreciate organizers for giving me such a wonderful opportunity to present our work as well as introduce our center in this meeting. So I am collaborating with Professor Andrea Agusta, which is the reason why I am here as a presenter today. So today, I'm would like to share with you our experience of the drug development as a rapid response to outbreaking, emerging and re-emerging infectious diseases, mainly focusing on the COVID-19 research. So if you have any interest or have some possible collaborating plan, please don't hesitate to contact me after the meeting. So I am here in this email. Okay, so please let me briefly introduce about our institute. So the National Institute of Infectious Diseases is a Japanese national institute operated by Ministry of Health, Labor and Welfare, Japan. So we have 24 departments and centers divided in the 3 branches. One is in the Toyama branch in Shinjuku, which is downtown Tokyo, where I am here, and 2 other branches at the suburban area in Tokyo. So our function includes the control and the testing of vaccine and biological products, surveillance and analysis, and the reference service for infectious diseases, and the international cooperation activities, and also the basic and applicational research. So to achieve these functions, we have these departments and centers, some of which is in charge of the research of specific pathogens, and some of them involved in the surveillance and the epidemiology and the regulatory science and the quality assurance. And actually, we are studying about the many infectious diseases using the specialized techniques. So we are here, Research Center for Drug and Vaccine Development. So our center is actually one of the newest center in our institute, but one of the biggest organization in our institute. We have 10 laboratories divided by Division of Vaccine Development and the Division of Drug Development. And I am in charge of organizing the Division of Drug Development. So the vaccine group is studying about immunology and the vaccine, and the Division of Drug Development is studying about the virology, bacteriology, and drug development. So here we have currently 7 staffs and also the 3 postdocs and a couple of visiting researchers and 10 students. And yeah, so Dr. Hairman Azam is originally from Indonesia, who is the faculty staff in the laboratory 10, who is studying about the phage therapy against antimicrobial resistant pathogens. So we are very welcome for any collaboration and exchanging program for students or researchers and some joint grant application or something. So please let me know or contact me, anything. So our target is the pathogens currently epidemic in Japan, such as hepatitis viruses, coronaviruses, and antimicrobial resistant pathogens, and also those potentially causing outbreaks in the future, including the insect-borne respiratory zoonotic viruses. So we study about pathogens such as the establishment of infection model system, the analysis of the mechanism of infection and pathogenesis, and the development, identification of new drug targets. And we also study about the drug development, such as the discovery or optimization of the antivirals toward clinical trial, and the development or optimization of modality, and also the automatizing the process for drug screening. And we also encourage collaboration with the academic, industrial, clinical, international partners, and is involved in the rapid response to the outbreaking emerging infectious diseases. And we train many young researchers as well. And in addition to the usual research activities, we are involved in the response to emergency related with the emerging infectious disease. So our original research is about the hepatitis viruses, actually, especially the blood-borne hepatitis B, C, and D viruses using the virological approaches. So I originally study about the mechanism of virus infection and also the drug development. So, for example, we established the infection model systems that can evaluate the hepatitis virus replication and identify antivirals that inhibit the infection or replication of viruses, and analyze the mode of action of these drug Cs. And we also analyze the molecular target of the antivirals. Here is an example of the NTCP structure, which is the entry receptor of hepatitis B and D viruses, recently published in Nature, and under the collaboration with other universities in Japan. So we have some skill of virus infection experiment, and also we have the platform of chemical screening analysis. And actually, I graduated from the Faculty of Pharmaceutical Science in Kyoto University. So my background is the pharmaceutical sciences. And also, we do have the collaborations with interdisciplinary field. Yes, so this is my background. So from here, I will talk about our research content, mainly about the coronaviruses. So, the COVID-19 induced or is inducing huge damage on the public health, the social and the economic aspect, having more than now currently 500 million confirmed cases worldwide. So for the response to outbreaking emerging infectious diseases, you need a public test system, surveillance and epidemiological analysis, as well as the development of diagnosis, vaccine, and drugs. So in our institute, each center or department have their own research resourcing put together to against for these issues. So, for example, the Department of Virology 3, which is specialized in the research about the respiratory viruses, has established a public test system in Japan in early days after COVID-19 pandemic, and other the Center for Surveillance Immunization Epidemiological Research and the Pathogen Genomics Center together was involved in the surveillance and epidemiological analysis. And the Department of Pathology has established the diagnostic system against COVID-19 in the early days after COVID-19 pandemic. And as a center also study about the vaccine, and we are in charge of the development of drugs. So this is the situation in Japan in 2020. So we have the first wave in Japan at April 2020, which should be earlier than the case in Indonesia. So yeah, so our institute identified the first COVID-19 case in Japan in the middle of the January 2020, and 2 weeks later, our institute isolated the SARS-CoV-2 virus using the cell culture system, and after that, we are involved in the response to COVID-19 after that. So I was decided to join the COVID-19 response team at the late January, and after BSL-3 room arrangement and training, actually we started the infection experiment at early March of this year. And after that, we prepared and opened the first paper in the preprint bioarchive at the middle of the April, which is 1.5 months after starting the experiment. And through the discussion with the clinical researchers in university hospitals, we have studied the clinical trial of one of the identified drug to the COVID-19 treatment. This is the time schedule. And now I talk a little bit about our experimental experience. Sorry, this is written in Japan, but in January, the Department of Virology 3 have isolated the virus from patient swab and saliva. So as you know, the COVID-19 testing is here. So the RNA of the virus is isolated from swab or saliva from infected patient to test by real-time PCR. And you have the negative or positive result, right? But when you isolate the virus itself, you cannot isolate RNA because the RNA isolation disrupts the virus itself. So you directly treat swab or saliva to the cultured cells that is susceptible to virus infection. So after 1 to 3 days, when you observe the cells, you can see the fusion of the cells from one to the neighboring cells, because the virus proliferation induced the cell-cell fusion. So this is one clue of the virus infection. And when you stain the cells with the antibody against the SARS coronavirus protein, you can see the detection like this. And electron microscopic analysis showed this kind of the particle in the culture supernatant having the very remarkable spike, which is a hallmark of the coronavirus. So now you can see the virus proliferate in these cells, and you can have the virus in the culture supernatant of these cells. And yeah, actually the Department of Virology 3 did this assay in only 2 weeks. This is very fast in our feeling as a researcher. So the point is that one researcher is studying about the MERS coronaviruses for a long time. And he knows that this cell line named Vero E6 TMPRSS2 is very susceptible to coronavirus infection. So using this cell line unable to proliferate the SARS-CoV-2 very well. And yeah, this is a very point for the rapid isolation. So after that, I did the drug screening using this cell culture system. So as you know, the development of drugs takes a very long time from the basic to the preclinical, clinical trial, from the identification of seeds compound, followed by the optimization and to be validated in the activity and the adverse effect in the clinical trial. So it takes generally more than 10 years. But during the pandemic of COVID-19, you cannot wait for such a long time. So one of our approach is to try to rapidly identify the drug candidate by drug repurposing approaches. So, we have more than 3,000 approved drugs against any diseases such as hypertension, or allergy, or anti-cancer, or immunosuppressive, or anything. So you may have the drug that happen to suppress the viral replication. So what we did at first is to screen these approved drugs for identifying those having the anti-SARS-CoV-2 activity. So this is a drug repurposing strategy, as well as development of new drugs. So we have constructed a collaborating system for providing the compounds from many university or institute. And we also have the collaboration with researchers in the field of biochemistry, informatics, and mathematics, and clinical medicine for the analysis. And after screening the compounds, we propose drug candidate to the clinical side as soon as possible. And after clinical trial, during the clinical trial, we also involved in the testing of the SARS-CoV-2 viral load in saliva to evaluate the effect of antiviral drugs. So this is the construction of our collaborating team. So this is the drug screening. So we use the SARS-CoV-2 isolate, which is isolated from a patient in Japan, from Wuhan. And after treating the virus to the cells, virus susceptible cells called Vero E6 TMPRSS2, actually you can see cell death by observing the microscope, because the proliferation of the virus produced a large amount of the virus protein that kills the cells by the ER stress or something. So our strategy is that treating drug library here. And if you find out some drugs that protect the cells from virus-induced apoptosis, this drug may be the that suppressing the virus infection or proliferation. So this screening is very easy because you only see the microscope. That's it. So we did the screening using this system. And finally, we automatize the observing of the cells by using the high content imaging analyzer. And this system enables a large number of the screening. But what I introduce you here is the first small scale screening using about only 300 compounds. And by this screening, we identified 5 compounds at first. So one of these, the nelfinavir, is another is cefalantine, which is originally the anti-HIV and anti-inflammatory drugs. So at first we confirm the antiviral activity by staining the viral protein shown by red here in infected cells. So when you treat these 2 drugs, you don't see anything about the viral protein expression, showing that these 2 drugs is effective on suppressing the production of viral protein. So we treated the different concentration of the drug at the x-axis here to see the viral RNA level by real-time RT-PCR analysis shown by y-axis in the logarithm titer here. And when you look at the 90% inhibitory concentration, that of the nelfinavir is about 1 micromolar, and also 1 micromolar for cefalantine as well. And when we compare the IC90 with those of the drug candidate at that time, expected drug candidate, such as the remdesivir, favipiravir, cyclophosphamide, hydroxychloroquine, or lopinavir, you can see the IC50 of 2 drugs were rather lower compared with these drug candidates. So showing that the activity against the virus is rather higher than these expected drug candidates. So we're focusing on these 2 drugs. And by the virological analysis called the time of addition assay, I don't explain about this assay very precisely because of this is a little bit complex. I just show the result. So this assay suggests that the nelfinavir suppressed the viral replication inside the cells, and the cefalantine inhibited the viral entry to the cells. The mode of action seems different. So what is the target protein of these 2 drugs? So I happen to know that other researchers in Japan screened in silico using the main protease of SARS-CoV-2 that has been opened in February in 2020, and he identified actually nelfinavir that could bind to the main protease of SARS-CoV-2. I happen to know this. So we identified nelfinavir by the viral infection assay, and they identified the same compound from the in silico analysis. And our biochemistry collaborator actually showed that the treatment with nelfinavir dose-dependently inhibits the enzymatic activity of main protease of the virus. So the mode of action is this. So nelfinavir bind to the main protease, which is essential for the viral replication inside the cells to inhibit the viral replication. So the mode of action of cefalantine seems to bind to the spike protein on the particle surface, which is the in silico analysis also by our collaborator. And our experiment supported this hypothesis, showing that treatment with cefalantine inhibits the interaction between virus and the cell surface significantly. So because the mode of action is different, we treated both of two drugs in combination and found that these drugs have a synergistic antiviral effect shown by orange in large area of the concentration range, showing that the combination therapy is likely to be more effective than mono treatment. So this is our lab work, but the issue is, are these drugs really effective in clinical settings? So before getting to the clinical trial, we predicted the antiviral effect by mathematical analysis, because we already have the pharmacokinetics information of these approved drugs. So for example, the Cmax of the nelfenavir in HIV patient is 4.5 micromolar in plasma with the 3 to 5 hour of the half-life. And we do have the dose-dependent curve using our infection assay. And this concentration seems to be to exhibit the antiviral effect. Yep. So our mathematical modeler predicted the antiviral activity in the clinical drug concentration and found that the cumulative viral load were decreased to less than 10% by nelfenavir, cefalantine, and the combination treatment. And they also predicted these drugs shorten the period until virus elimination by 4 to 6 days. So with this information, we discussed with a clinician at Nagasaki University Hospital, actually, and they selected the target patient and established a clinical protocol. And this clinical trial has started in July of 2020. And after that, we tested the viral load by real-time PCR in the saliva from the patient and get a feedback to the clinical site. So this is a study, a clinical study for nelfenavir, actually, having the 10 hospitals in total, so randomized multi-center trial. And we prepared this paper of the infection assay and opened this paper in the preprint server bioarchive in the middle of April. And because of the limitation of time, I don't mention about this, but other drug candidate, mefloquine, a clinical study is now underway in Egypt. So this is an international collaboration as well. And we also did the screening from 3,000 and approved drug to get 10 drug candidate, and we are now under analysis also. And so from the drug repurposing approach, we identified some of the drugs, but in my feeling, the antiviral activity is relatively mild, not very high actually. So yeah, this is okay. And we expect the effect in the clinical trial, but we also need to have new drug of which activity is super strong to control this COVID-19 for a long time. So we did this kind of the collaboration with more than 50 collaboration projects from different modality, not only small molecule, but also peptide, nucleic acid, natural product, and others. So this is one of the example. We have a really nice opportunity to have a collaboration with Professor Andrea Agusta and Kami Takahara Sensei at Kyoto University, and Professor Maehara at Fukuyama University, and Tanaka Sensei at Nagasaki University as well. And from the 67 compound from Indonesia, we actually identified several compounds that reduce the viral RNA level, although it is still very high concentration. So now we are analyzing these compounds as a possible new drugs. We are expecting this project as well. Okay, so we did many things, and sorry, this is most of these are written in Japanese, and we found some candidate from approved drug or new drugs, and some of them are in the clinical trial. So, but really approved drugs worldwide is this one, remdesivir and the other one. So when you look at these drugs, the dose shown by blue is the drug repurposing approach, actually. So remdesivir has been a candidate of anti-Ebola virus, and dexamethasone is a steroid, baricitinib is an anti-rheumatoid drug, and molnupiravir and nirmatrelvir is a drug candidate against originally hepatitis C and the SARS virus that is epidemic in 2003 in Hong Kong. So, but many of them is not really approved, but the drug from drug candidate, such as remdesivir, molnupiravir, and nirmatrelvir. Yeah. But two other drugs are really new drug established from after the pandemic of COVID-19. So these are the antibody, different modality. So as you know, the development of small molecule takes long time, but the development of antibody is very fast, very rapid. But the limitation of the antibody is that the target is limited to the extracellular side, such as spike, and also only injection. And we have other limitations as well, but very rapid. So from now, you need to select modalities depending on the characteristics of infectious disease or the needs from society, how fast we have to develop. So we also have the peptide or nucleic acid as new modalities. This is between new small molecule and antibodies, I think. So we need to focus on peptide and nucleic acid as well. Okay, so I have 2 or 3 slides. This is the last part. So how we have to be prepared against emerging and re-emerging infectious diseases. This is a problem. So when you look at the history of the outbreak of infectious disease, all of them are RNA viruses, either plus or minus strand RNA virus. And many of them are respiratory, zoonotic, and insect-borne. So these viruses maybe have some potential to have the outbreak. So I thought that only RNA virus we have to prepare. Yeah, I thought that, but the answer is no. This is a reality. So this year we have the monkeypox outbreak in many of the countries, now from the May of this year. And this is actually the DNA virus related with the smallpox. And this is, yeah, zoonotic, but not respiratory in at least in our understanding so far, and not insect-borne. So it is very difficult to predict which virus will have the outbreak. Yeah, and this monkeypox causes a human-to-human transmission by originally the contact infection, but currently also through the sexually transmission. And there is no FDA approved vaccine and drug, but we do have the vaccine and drug against smallpox. So now we expect these anti-smallpox vaccine and drug may be effective on monkeypox. That is our current standpoint. But in a paper published in Lancet Infectious Disease in May of this year shows that BRIN cidofovir, which is an approved anti-smallpox drugs, has no clinical benefit, but rather significant liver damage that resulted in the treatment cessation. So yeah, we may need another really antiviral drug against monkeypox. So we did that from approved drug library and found a couple of drugs, including the adalah. Yeah, we did this kind of approach and opened this paper in bioRxiv and early of this month. After the 2 months after deciding the rapid response. So this is the last slide. So now our infection assay pipeline is expanding from not only plus strand RNA virus, but also minus strand and DNA virus as well. So the response to emerging virus is a global matter, yeah? And establishing the international collaboration is very important, I think, and training researchers is very essential to have a good response to these infectious diseases. So yeah, I am really welcome for the collaboration, the exchanging program and joint grant application with you. And I expect to have some relationship with you in the future. Yeah. Oh, this is advertisement. I will organize the International Hepatitis B meeting next year in Japan. So if you have some interest, please join us. Thank you very much.
Thank you very much, Professor Watashi. I would like to welcome all any questions from the participants. Okay, first maybe from me, Professor Watashi. You mentioned that you have the around 3,000 screening of the already known drugs. What is the differences previously mentioned for nelfinavir and cefaratin screening, but also the international screening drug candidates for mefloquine? Actually, how to make the differences between the two types of screening for the Japanese screening, Japan availability drug screening, or for the international one?
Yeah, yeah. Thank you for the question. So initially, actually, it takes time to screen as much as 3,000 drugs by infection assay. So initially, what we did is to screen the small scale of the library consisting of about 300. That is that consists of mainly the antiviral, anti-fungi, and immune-related approved drugs, because in our experience, these kind of the approved drugs have a high percentage of the antiviral drug in any of the new viruses. So we have first priority of this kind of the drug library, and we screen that and identify nelfenavir and cefalantine from this library. But after that, finally we have to screen all of the approved library available currently, and these 3,000 drugs screening we studied after identification of nelfenavir and cefalantine, and found 10 other drug candidates. So this is our story.
So and then you can how to say directly use the two candidates for the clinical trial. Is that correct, Sensei?
Yes, yes, and yeah, the nelfenavir and also mefloquine, which we identified after nelfenavir and cefalantine, is under clinical trial in Egypt, and about 10 other approved drug we haven't do the clinical trial about this. And also 2 drugs are under clinical trial from new drug, not approved drug. Yeah, this is our what we did so far.
Actually, Sensei, I'm also curious. You mentioned about the high content imaging analyzer. So can you explain us more about this typical for screening? Yes.
This one,
because I think this typical screening is one of the method for faster.
Yes, exactly. So
yes.
Initially, what we did is to observe the cell morphology manually by our eyes, like her in this lab. But it takes time as well. So, so we established the system for counting, automatically counting the cell number by using the high content imaging analyzer. So what we do is to fix the cells by paraformaldehyde and treat with DAPI for nuclear staining. And this machine count the number of the nucleus in one well. So this is what this machine did. And yeah, this system save our time actually. Yeah, so if you have to screen, I would say more than 1,000 compound, it will be better than the manual observation.
So, Sensei, I also would like to ask, so how long it takes from, can you rewind again the screening started from the screening until you can get the clinical trial improvement?
Yeah, yeah, we started the infection experiment from early March, and the clinical trial for nelfenavir has started in July. So it takes four months.
4 months only.
Yes. So actually, we prepared the first paper in April, and after that we have this discussion to clinicians about the plan of the clinical trial. And after that, you know, the clinician has to be approved for IRB in the hospital. And also they have to get a scientific grant for the clinical trial. Yeah, so they did these things in May and June. And finally, the clinical trial started in July. That's, yeah, what we did.
Very remarkable, Sensei. And I would like to read, Sensei, if you don't mind, some of the questions from the chat box.
Yep.
The first one is from Niputu Ermi, asking about for testing new compounds or new candidates as antiviruses. Is it possible to get a clinical fit without going through in vivo testing? In addition, that's the discovery from for the new drugs in the
new
in
drug.
vivo. Yes.
I don't think you can do that without animal infection experiment,
Yes.
because yeah, we have many information about approved drug, including the effect and the adverse effect. So yeah, if the clinician think this drug is safe, they may get into the clinical trial without enough information about the antiviral effect in the in vivo animal model like nelfenavir. But new drug, you don't have many information about that. The most important thing is the adverse effect of the new drug. This should be essential. So, and also, because there is not much information about the new drugs, eventually, yeah, you have to evaluate the antiviral effect that in vivo infection experiment. We did that about one drug that is currently under clinical trial. So yeah, should be essential to do the animal infection experiment and the pharmacokinetics and also the toxic experiment. Yeah.
Another addition for the question, Sensei, the discovery for new drugs in vivo assay, both enzymatically and cell culture, not required for the positive control, or how to believe the result that if we can get the really potential new drugs?
Yeah, we always put some positive control in all the experiment. So for example, although I don't put here, we always use hydroxychloroquine as a positive control in infection experiment, although the hydroxychloroquine was not effective is in the clinical trial, but it is very effective, at least in the cell culture assay. This is already reported in many papers. So we put some positive control here. And also in the in vivo infection experiment, we put molnupiravir as a positive control, which is very effective in vivo infection experiment. So yeah, you need to compare the antiviral effect or some result side by side, to your positive control. Yes.
We'll go on to the next question from Professor Andrea. He said that thank you very much for the presentation, Watashi Sensei. And then from your research and clinical testing, it's already found 2 compounds that can be reduced the infection of virus into cells. Is the combination drugs already used to treat the COVID-19 patient in Japan?
No. Yeah, this is very difficult to do the clinical trial by combination treatment from the first. So yeah, it is desirable to do the combination treatment from the first, but yeah, things is like that. They need to have some information about monotherapy. And if you have some effect of monotherapy, they think about the combination treatment. Yeah, unfortunately, this is the situation. Yeah, yeah.
So you mean that the clinical trial is for the combination is still very long to understand?
Yeah, needs longer time, I would say. Yeah, not only for COVID-19 drug, but also other drug, unless one drug company develop other types of the drug simultaneously, like Gilead Science develop sofosbuvir and ledipasvir for hepatitis C, and they put together for combination and do the clinical trial from the first, because they have multiple drug candidate having a different mode of action.
So there is another question in the chat box from Bapak Wempi. Dear Mr. Watashi, multi drugs always making more advantage to make more activity drugs for the disease. I think multi drugs will make more hepatotoxic and renal toxic, isn't it? And would you mind explain about it? Thank you.
About the toxicity against the liver and the kidney.
For the combinational drugs that previously mentioned.
Yeah, it might be. Yeah. So yes. So eventually you have to check the adverse effect for combination treatment and also mono treatment, of course, especially for the new drugs.
So we'll go on again, Sensei, to the next question from Dr. Aswad. Very nice presentation. And did you try to combine the two promising compounds with other approved COVID-19 treatment? For example, like remdesivir to obtain the optimal antiviral effect.
So yeah, combination of nelfenavir and remdesivir also synergistically exert the antiviral activity. Yeah. So at least in the cell culture infection assay. Not in the clinical, not in the in vivo assay. So this is what we have as a data. So but after that, we didn't do anything about the combination of remdesivir and nelfenavir for the clinical trial, because as I mentioned, the clinical trial for combination treatment is very difficult.
Can you tell us a little about the result, Sensei? Whether the combination will affect as an antivirus for after the combination?
Yeah, so in the cell culture infection assay, yeah, you can see a synergistic effect of nelfenavir and remdesivir, and also nelfenavir and other approved drugs such as molnupiravir. So yeah, you can see that. But the problem is that even if we imagine that the combination treatment is desirable compared with the mono treatment, the systematically something difficult to do the combination treatment in the clinical trial, at least in our country, in our system. That's one of the barrier that I am feeling.
Another question from the chat box. from Mr. Danang Waluyo. Thank you for the presentation. During screening, you also tested some of natural products. Based on your experience, how does the potential of those natural products as source for screening of antiviral agents? And do you think natural products are good source for the screening as fast as the known, compared with the known drugs before you mentioned in your presentation?
Thank you. Yeah, I like natural products. Yeah, honestly, because because natural products have a very diverse activity, as you know, because the structure itself has very diverse. And now what we have authentic antiviral drugs targeting the enzyme. So the protease or polymerase or integrase or neuraminidase. But because these drugs is analyzed very well so far, we have to move to develop the inhibitor of protein-protein interaction that is essential for virus infection or proliferation, such as the binding of spike with its receptor or something. So by inhibiting the protein-protein interaction, you need a molecule that is bigger and having a large surface in its molecule. So in this point, the natural product is, I think the natural product have a very good candidate for the drug, for the infectious virus drugs.
So in correlation, I think about the natural products questioned here in the last question in the chat box from Mr. Agus Winarso. Do you have the research any about the herbal of Sambiloto as a traditional medicine to combat the COVID virus? Can the marker drug can kill the virus?
So far we have several collaborating projects related with the natural product, and we found some natural product have a high activity against SARS-CoV-2 infection or replication. Although in many of the cases, the mode of action is very difficult to analyze because we don't have much information about what is the target of such a natural product. So it takes time to analyze the mode of action, but the activity itself sometimes very high. The reason which we don't know currently. Yeah. So we expect to be a good drug candidate from natural product library. Yeah.
So thank you very much for the speech, Professor Koichi Watashi. This the end of the first session. Professor, you can, I heard that you have another appointment. You can leave the Zoom meeting if you would like to attend the appointment. Thank you very much, Professor.
Yeah. Thank you very much.
So the next session is from the second speaker, from Professor Dr. Makoto Hashimoto, the topic about the photo-affinity labeling to elucidate functional analysis of ligand biomolecule interactions. Professor Makoto Hashimoto is a professor at Graduate School of Agriculture, Hokkaido University, Japan. he got his bachelor, master, and PhD in pharmaceutical science in Hokkaido University, which the main topic is about the photo-affinity labeling with biotinylated phenyldiazine. You can see from the screen his all current publications. I'd like to ask whether Professor Hashimoto already in the screen. Can you share the screen first, Sensei?
Okay, sorry, Sosa. Please wait.
Yes. Actually,
Okay.
yes, I can. Yes, I can clearly see your presentation.
Okay.
So Hashimoto Sensei, time is yours.
Okay, so I will start. Thank you for your kind introduction. I am Makoto Hashimoto at Graduate School of Agriculture, Hokkaido University, Sapporo, Hokkaido, Japan. I'd like to thank all organizers of this conference for giving me these opportunities. Hokkaido Prefecture is located at the north end of the Japan and one of the 4 major Japanese islands. Sapporo City is the capital of Hokkaido and located 800 kilometers of Tokyo. There are about 2 million people in Sapporo City. The Hokkaido University is established in 1876 and has 18,000 students, including the 2,000 international students from 134 countries. Hokkaido University has a liaison office in Bogor, Indonesia. The academic relationship between Indonesia and Hokkaido University is very close. This slide summarizes today. I will present each part in this order. First is what is photo-affinity labeling. Second is photoforwards properties of the construction. The third is photoreaction in diluted sample. Fourth, combination of photoforward and tag to elucidate labeling site. For photo-affinity labeling experiments, first, the researchers prepare the chemical compounds which consisted of the ligand and photoforwards. The target biomolecules recognize the ligand derivative and forms the complex in a reversible manner. After making the complex, high reactive chemical species generated by photoirradiation. The generated chemical species make covalent bond between the ligand and biomolecules. At this stage, researcher can recognize the ligand-utilized biocomponents. Further investigation of the cross-linked component can reveal cross-linking site in the target molecules. These information will be utilized for the molecular mechanism of ligand recognition by biomolecules. This slide shows brief summary of the main method to understanding the mechanism of molecular interactions between small bioactive ligands and biomolecules. Genetic approaches provides an efficient and indirect route for pinpointing functional amino acid within biomolecules through deletion or the mutation of the native amino acid alignment in a proteins. Spectrometry approaches, which are 3D structure determinations involving the NMR spectroscopy and homology modeling are a powerful and direct approach for the analyzing the 3-dimensional structures of proteins and at the atomic level. These approaches will be suitable for the purified biomolecules. Chemical approaches which are fundamental in chemical biology, provides an alternative route for the direct identification of the target protein in biomolecules, as well as the ligand binding site structures. Because the analyses are based on the affinity between the ligand and the target proteins. This slide shows what kind of the fundamental fields will be needed for photoaffinity labeling. Selection and synthesis of photophores containing ligands will be based on the organic chemistry. After the cross-linking, the analysis, the cross-linked component will be based on the biochemistry. So it is slightly difficult to tune up the methodology or all of the including of these fields. It is important which photophores are used for effective photoaffinity labeling. In nowadays, typically allyl azide, benzophenone, and allyl diazolene are used. Allyl azide are photoactivated below the wavelengths of the 280 nanometer, which sometimes causes damage to biomolecules. And after the irradiation, the generation of the nitrogen as active species, which sometimes rearrange to ketamine, making the 7-membered ring and as undesired side products. Benzophenone are photoactivated with light over the 350 nanometer and generate the reactive biological states. These regenerate ground state carbonyl compounds, so benzophenone are reusable for photoaffinity labeling. Although these photophores sometimes use long photo irradiation times for photoaffinity labeling. The third one is allyl diazolene, are also photoactivated with light over the 350 nanometer and the generated carbenes, which are more highly reactive species than other photophores. So the rapidly forming cross-links to biomolecules with short photoirradiation times. It has been reported that the photolysis of the diazolene can cause the diazoisomerization, giving undesired intermediate in photoaffinity labeling. Diazoisomerization can be suppressed by the introduction of the trifluoromethyl group into the diazoyl ring. Comparative irradiation studies of these 3 photophores in living cells suggested that the irradiation needed for the generation of active species from azide and benzophenone causes sometimes cell death because the long irradiation time are needed. Although these results indicate the photoaffinity labeling with trifluoromethylphenyldiazolene, allyl diazolene has the most promising nature for photoaffinity labeling. But the applications are not so many as with other photophores. Because the synthetic construction of the 3-membered rings, diazolene, is not easy as with other photophores. Synthesis of the diazolene 3-membered ring required at least 5 steps from the corresponding allyl halide derivatives. The 5 steps will be needed. A substitution group should be introduced onto the benzene ring to connect to the ligand skeleton. The previous synthesis for the diaziline is a diaziline derivative have pre-installed of the substitution group before the construction of the diaziline moiety. The repeated construction of the diaziline moiety for each derivative is a drawback to apply the photophore for Auto-Affinity labeling. So we encourage to archive so the post-functional synthesis for the trifluorophenyl diaziline. The mother skeleton diaziline derivative will be prepared as a large scale. Then the substitution group introduced to mother skeletons by organic synthesis. For example, so the other substitution group can be available. So this is one of the major the mother skeleton. The metamethyl substituted diaziline is selected as a mother skeleton because first is a methyl group. Strongly activates for the nucleophilic aromatic. Easy to introduce electrophile. Second, its orientation of the substitution favors of the ortho position of the methyl group. And the third one is if the demethylation of the methyl group will be easier than that of the para isomer, and the generated the alcohol will be utilized for the alkylation and or the acylation are utilized for the introduction of the tag. Compound H, mother skeleton of the compound H smoothly underwent the introduction of many subsequent group in the usual synthetic manner without the decomposition of the diazinyl ring. Although the methyl group was helpful in the aromatic substitution reactions, it sometimes causes reduced biological activity in the target biomolecules. We also attempt post-functional derivatization for the unsubstituted diazinyl 2 under various conditions. The diazinyl moiety acts as an electron withdrawing group than its substituted counterparts. But it was reported that the diazinyl ring was easily decomposed in the presence of strong acid over the 25 degree. One of the reactions, so the dichloromethyl ether and titanium chloride in dichloromethane, which is one of the formulation conditions for aromatic compounds, was useful for the methyl substituted compound 1 with moderate ease, 65%. The reaction can be improved in the neat condition up to the 85%. But the compound 2, unsubstituted compound 2, cannot apply. So the titanium chloride and dichloromethane, dichloromethyl ether and in dichloromethane cannot be applied. So we tune up this reaction to utilize trifluoromethan sulfonic acid. The condition with trifluoromethan sulfonic acid without solvent is easily able to afford the formulated compound without the composition of the diazinyl ring at 0 degree. It is my first time to utilize TFOH, which now Zetri will keen to utilize. TFOH and is not only strong brain state acid, Also, high solubility for many organic compounds, even though the alpha amino acid skeleton. Homo and bis-homo phenylalanine derivatives, which is longer methylene part in the side chains. Previous and synthetic route is Friedel-Crafts acylation of the aspartic or the glutamic anhydride in the presence of large excess of aromatics, with aluminum chloride under the reflux condition. The reaction uses a large excess of aromatics, so it is not suitable for the precious phenyldiazine compounds. So I try to tune up of the reactions. The reaction, so I use the TFOH as catalyst and solvent to proceed the reaction. Equivalent of the acyl acceptor and acyl donor of the alpha amino acid in TFOH became the homogeneous mixture, and the Friedel-Crafts acylation proceeded at 0 degree within an hour. So after the reduction, so the very high yield, so the homo and bis-homo phenylalanine derivatives can be obtained. The established condition apply to the diazine compound 1, same as before. Equivalent diazine and alpha amino acid derivatives in TFOH afforded the diazine, the homo and bis-homo phenylalanine derivatives in moderate yield. The post-functional method is also utilized diazine indole derivative. From the diazine indole derivative 3, the biological active indole derivative are synthesized. Next is a photolysis of the diazine photophore in diluted solution. In organic photochemistry, it was reported that diazine A was photolysed with a black light lamp to generate corresponding carbene and diazoisomer B. The diazoisomer B is easily generated carbene and on use of the shorter wavelengths 302 nanometer or with longer irradiation with black light. The photolysis experiment in organic photochemistry is conducted high concentration of solution, typically over the 1 molar. On the other hand, actual photoaffinity labeling for biomolecules were performed at less than 1 millimolar. So compound A, B, and C can be distinguished on 90F fluorine NMR studies. Proportion of the photodecomposition mixture at 1 millimolar of compound A in solution with with black light lamp revealed that diazoisomer B is not a serious problem over the 10 minutes irradiation. But if the compound A started from the 10 millimolar, so after the 10 minutes, the 30% of the diaz compound was observed. But this 10 millimolar solution was diluted to 1 millimolar. And after that, then the irradiated the same light, same black light, so that it is smoothly within the 10 minutes, so the generate of the carbene. So ensure the previous results were consistent with actual photoaffinity labeling. L-amino acid oxidase with 10 micromolar of the geazinyl phenylalanine derivative, and chymotrypsin with 200 micromolar of the phenylalanine paranitral derivatives are conducted photolysis at 354 nanometer only, or the with or the without 3O2 irradiation. There were no difference between the irradiation 305 nanometer only and the irradiation at 350 nanometer followed by 302 nanometer. From these results, the photolysis of the diluted sample will be different from manner of organic photochemistry. So even though the photoaffinity labeling condition was set up as described before, photoaffinity labeling efficiency for the generation of the covalent bonds between photophores and biomolecules was always less than 10%, very low yield. So the special treatment for cross-linking component is needed. I select the specific interaction between abigine and biotin is utilized for this purpose. Abigine protein recognizes a small molecule biotin with a very strong affinity. This specific interaction has been combined with photoaffinity labeling. We so-called the photoaffinity biotin relation. The compound consisted with photophore, theta-2 ligand, and the hydrophilic spacer and the biotin is synthesized based on the post-functional synthesis. The abigine biotin complex was detectable by chemiluminescence detection with the same sensitivity as archived by radioisotope analysis. So this compound was utilized for the biotinylation diazine compound is applied to the functional analysis beta-1 for galactosyl transferase. The enzyme utilizes UDP galactose and donor and the N-acetyl and glucosamine as acceptor to generate N-acetyl lactosamine. Carbohydrate transferase are low homology and crystallization are not easy to functional analysis. The N-acetyl glucosamine asparagine skeleton, N-acetyl glucosamine and the N-acetyl the asparagine skeleton is modified with biotin-related geazinyl photophore as compound 4. Photoaffinity labeling with or without competitive inhibition with N-acetyl and glucosamine are conducted. The enzyme utilizes two substrates, donor and acceptor, so the experiment were also conducted with or without donor derivative of the UMP. The specific photolabeling efficiency depends on the temperature manner. The high temperature is high specific, and with the UMP coexist of the UMP dependent manner. That indicate in the presence of UMP and the temperature, that may indicate the photoaffinity labeling can reflect molecular dynamics of biomolecules. The cross-linked peptides are recovered by abigine-biotin interactions and identified as cross-linked amino acid as methionine 225. Combination with the results and the in silico analysis indicates to the reveals to the carboxyl group of the aspartic acid 418 make critical role activation of the N-acetyl glucosamine for hydroxyl group for the efficient and galactosyl transfer. We developed the photoaffinity biotinylation of the glucose transporter as a typical experiment for living cells. Glucose transporter proteins are the particular importance as a pharmacological targets, and many transporters are membrane protein for maintaining homeostasis. Among the transporters, GLUT4 proteins is a particular importance as it is present only in insulin-responsible tissues. In the basal state, the transporter is sequestered into the intracellular reservoir membrane component. If the insulin signaling by stimulation, the exocytosis the GLUT4 containing vesicle on the cell surface. Cells were photolabeled at exofacial stages. Then the labeled transporter can be analyzed to reveal the sequester's mechanism for the transporter, because this one is endocytosis. This type of analysis is not easy by conventional biochemical approaches. Previous studies reveal that the C4 hydroxy position is in hexose tolerate the introduction of the bulky substituent to provide substitution for glucose transporters. Auto-Affinity labeling with the C4 modified hexose derivative is particularly acceptance of membrane glucose transporters. So I synthesize many of the compounds. Determination of affinity constant for biotinylation glucose analog with the varying spacer arm lengths, half maximum inhibition constant for the analog interaction of the glucose transport activity were determined in insulin-stimulated rat adipocyte. Also, the K1 value are increased in the spacer length manner. The compounds 7 to 9, which have longer linker, will be suitable to detect labeled glucose transporter on the cell. So the next one is a combination of the photophore and the isolate tags. This association of the labeled component from the abicin-biocin complex is sometimes hampered because the complex is too stable to disassociate. 2D electrophoresis is one of the major analytical methods for the proteomics studies. The combination of the isoelectric focusing and the molecular weight analysis promotes the analysis for protein with high resolutions. Isoelectric pH value are unique for each protein. Therefore, altering the isoelectric pH value of labeled component will facilitate their isolation from unlabeled component by isoelectric focusing. If the isoelectric focusing is changed by only the photolabeled component, it is very useful. So the polybasic photophore, the polybasic amino acid group can be used altering the isoelectric pH. The geagenyl photophore containing the epsilon Bock resin derivative effective resolution the labeled proteins by altering the pH value. These Bock group can be easily hydrolyzed under acidic condition. The photolabeled component or peptides are changed to the isoelectric pH. Then the migrated component are digested with Resil-C protease. So the labeled component will be back to the previous isoelectric pH. The chromatofocusing profiles of photophore 10 before and after the trifluoroacetic acid treatment. Without acid treatment, isoelectric pH is around the 6. On the other hand, with acid treatment, change the isoelectric pH to over the 7, becomes the 7. Further, each isolated samples are treated with trypsin digestion. Only the only the acid treatment sample was photolyzed by trypsin. Proteolyzed by trypsin. That means biotin moiety was completely hydrolyzed. So to detect the component, recently the bioorthogonal chemistry, which is utilized reaction of the unnatural functional groups, is one of the fashion for recent progress. This slide shows the utility of the Staudinger ligation, which react azide and tryphenylphosphine derivative with intermolecular amide formation. The diazenyl-benzyl azide derivatives modifies PBDF membrane and irradiate. Then the limited amount of the tryphenylphosphine, biogenylated tryphenylphosphine derivative react on the membrane. The detection of this scheme. So the detection limit is over 50 picomole amount of the tryphenylphosphine derivative, only the 50 picomole. The data shows chemical yields of a quick reaction with azide and The data shows chemical yields of the quick reaction with azide and alkene in the presence of the 2+ bromide. The chemical yield is drastically decreased in a concentration-dependent manner. If the one compound is utilized 7.5 millimolar, 7.5 millimolar sample cannot improve, so the large excess amounts on the other side compounds. These results indicate that it is very important. Concentration of the target molecules are very important for bioorthogonal reactions. So the recent progress in mass spectrometry has opened the way to the identify labeled components through mass experiment. Stable isotopes are very useful for the tags exogenous component of the labeled component in mass spectrometry. Synthesis of the stable isotope labeled diadenine derivative are easier than that of the radioisotope compounds. So we have to we utilize post-functional synthesis to introduce the stable isotope labeling. So I present brief development of photon-affinity labeling today. Just now, our lab has been developing photon-affinity labeling with sweet and bitter ligands to elucidate the gustatory receptors. So today's presentation is based on our several reviews and books. I would like to thank the many supervisors and the students, including Zetri, who have contributed to this research. Please do not hesitate to contact me if you have any further questions or concerns, including the study in Japan. Thank you for your presentation.
Thank you very much, Hashimoto-sensei. Very great presentation. We have, I saw 2 questions in the chat box. The first one is from Mr. Agus Winarso. He asks you about whether you can make the drug design and then detect the mechanism of the specific functional groups of diazerine to the for the COVID-19 virus treatment.
So it is theoretically available, but it is not easy at this stage. So the drug for the compound is not easy to obtain. So as first speaker, we have to collaborate about to analyze the functional group, analytical functional analysis.
So, but the the crosslink, it's the photo-affinity label also can be used for the typical antiviral drugs. Can you explain more about that?
So it is important why the labeling is so low. So lower and lower. So that one is a three-dimensional. So the structure will be important, and the introduction of the diazoline moiety will be sometimes hampered to the interaction of the ligand and the biomolecules. So we have to tune up the time by time. So the very many experiments will be needed at this stage.
So since I move to the second question, dear from Basit Febrian, dear Professor Makoto Hashimoto, thank you for insight about the affinity labeling. May I ask about the effect of additional bond from photo-crosslinker to the native properties of drugs specified to the protein receptor? What advantage of photo-affinity labeling compared to using only the fluorescence agent?
So typical one is glucose transporters. So the normal state, so that type of the compound is the endosome, endocytosis and in the cell. But when the use so the outer stage, so we can photo-affinity labeling. So the covalent bond will be, so if you can detect so the fluorophore on even though the endocytosis, there is no problem. But sometimes so the disassociate with the fluorophore, so the photo-affinity labeling will be the advantage at this cross-link to the transporter, cross-linked with transporter. That one is one of the major the advantage for the other compound. So fluorophore is fluorescence is if you can detect and fluorescence, it is enough. But sometimes so the disassociates that compound.
Still connect with the previous question. Is it feasible for radiopharmaceutical to utilize the concept of photo-affinity labeling?
So the radioisotope is one of the ways. So the handling is very difficult. So the limitation, it is many limitations for the isolation. So I do not present at this stage. So for example, in organic the synthesis, so if the normal stage without the radioisotope, there is no problem. So it is enough the chemical yields. But the tritium labeled compound will be less than 1/3 because the less amount of the reagent will be available.
So still limit about the yield also will be considered.
Yeah, that's right.
I will move to the next question, Sensei, if you don't mind. The next question is from Dr. Tenny. Thank you very much for the presentation. What the basic principle of photo-affinity labeling for the potential new drug discovery design? From your research, is there any significant differences between the compounds with photo-affinity labeling and before labeling in the terms of efficacy for the new candidate drugs?
So just now, so the many of the genetic, so the genetic procedure or some other procedure will be exist, but if it is not easy to identify so the target protein or the target the labeling site by easy way. So the genetics is the spectrometry is available. So that if the proteins of some others, the information will be available. But this one is the photo-affinity labeling is based on the affinity for the biomolecules. So sometimes the agonist or the antagonist, it is very easy to detect all of the interaction to detect from the photo-affinity labeling. So even though the mixtures in the cell or some other biomolecules, so we tune up this method from the organic chemistry to biochemistry.
Thank you, Sensei, for the answer. Next on from Dr. Aswat. Thank you very much for the interesting presentation. The photo-affinity probes bearing many parts such as linker, biotin, and etc. Besides using the competitor of the ligand, are there any way to make sure that the probe can take place in the binding site of the protein?
That's right. So that it is very very good and question. So that just now, for example, the combination of the photo-affinity labeling with bio-orthogonal the reaction. That's for example, this type of the standing or the click reaction is a very one of the way to solve the problems. The minimum substitution of the ligand compound. But I present is here, so the only the, for example, even the 50 picomole of the the reactivity of the bio-orthogonal reaction will be very limited. So that we have to care about the efficiency for the how to make the orthogonal is very important. Just now, our lab will be tuned at how to effective to the bio-orthogonal reactions.
So, meanwhile, waiting for another question in the chat box, I would like to ask you. I would like to move backward in the beginning. So, actually, can you tell us more about the how to synthesize the cross linker, and then also what is the difficulties to synthesize this typically small molecule attachment to for the labeling in the macromolecule system?
So that maybe that we can understand about that. So that, for example, the linear reaction, it is very the classical reaction, but the handling will be very exosomatic. So there is very difficult and difficult to handle. So furthermore, the liquid ammonia should be essential to construction of the diagenine. So just now, it is some reagent will be available, commercially available at this stage, but it is very difficult to the construct of the diagenine. So we have to essential to perform this type of the reaction will be easily.
So, Sensei, previously you mentioned that the diazurin is more advanced rather than two other typicals of the cross-linker. Can you explain more about it? Whether the other two types of the photophores is it difficult to synthesis or something?
So the generation of the reactive intermediate is reactivity of these intermediate is quite different. So the carbene is the triplet stage and the singlet stage, also both of the available. But biological is it is slightly difficult to cross link, and the nitrogen is very the isomerization of the nitrogen will be one of the most drawing back nature for this compounds.
So how to choose the correct props based on the differences of the three difference of these protocols.
So
for the
but I present in this lecture, so the allyl diagenin is best, but sometimes so the benzophenone, only the benzophenone derivative can be can have the biological activity. So until so the synthesis, it is slightly difficult to predict about which protocols will be suitable.
See, we are still waiting for the other question in the chat box. Okay, one more question, Sensei, from me. So, what is the wise approach to choose the correct path or way to synthesize the full props to be as a candidate for the correct photoaffinity labeling of the macromolecules? Can you give us some advice for this type of synthesis?
So for example, so based on the peptide ligand, for example, so the substitution of the phenyldiazeline of the tyrosine of the tryptophan is one of the way to it is very small the modification. It is a very small modification, but if maybe so the natural product has always has only the aromatic rings. So how to use aromatic ring will be one of the way to consider about the molecular modeling.
So I think it will be the last question from me, and I saw no question in the chat box also. So this is the end of your presentation, Hashimoto Sensei. Thank you very much.
Thank you.
Thank you. So I think this will be we will turn to the last session of our seminar for today. I'd like to introduce you to Professor Hanafi. Wait a second. The third or our last topic from Professor Muhammad Hanafi is about the drug discovery and development process from natural products as candidate drugs. Professor Hanafi is a principal researcher in Research Center for Pharmaceutical Ingredient and Traditional Medicine, National Research and Innovation. He graduated in bachelor degree in University of Indonesia, master and PhD in Natural Product Chemistry, Faculty of Science, Osaka City University in Japan. He won several awards in 2021, the Habibi Prize, and then the Encouragement Award, the Hitachi Global Foundation Asia Innovation Award in 2020. In the screen, you also can see his current publication. So, Professor Hanafi, can you share your screen?
It's fine. Just a moment. Can you see our presentation?
I'm still not yet see the PPT. Can you turn the full size on the on the right bottom? Yes. Still loading.
Great. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good afternoon. Thank you very much for the introduction, Dr. Setri and Dr. Tani Renati, to give me chance to presentation in this interested webinar. So actually, in this morning, you just heard from the book center from Japan. The first is, I think, how to screening of the compound, and then the second one, I think, from the development, the new methods for IOC maybe. So my title is still Journal Discovery and Development Process for Natural Resources, but actually my background is chemistry, so I would to focus on how to optimize the lead compound after we get from the natural product. Yeah, actually everybody know about this one. I just in the morning probably already introduced. Since I have many mega in number 2 in the world, and I think we also still have the problem about the transnational, and the product is still mainly limited in the market. But also, and then the more than 50% raw material, drug, and also chemicals still imported. And the other one, the process of new drug discovery and development is still complicated and high cost and risk and development. And I think you need more variety scientific or more than maybe around 100 scientists with different competency. For this one, I think you need to get the lead compound. I think you can easy to isolate major compound is possible or other compound, and the starting design and development to optimize the activity. I think the moment in this morning you have the person. I think this day is very focused on the research target, and I think you know, and you said everything is limited. I think you must make cooperation also heard from the what sensei is also still need of our collaboration. But the problem is for this target, we need strategy for structural modification lead compound for candidate drugs. Yeah, this one is step for the research phase in the drug development. Actually, from 510,000 compound, only one we can drug in the preclinical. Only we can get the 25 to 50 hundreds compound, and then 5 compound in clinical trial, and only one drug will become. Maybe in general, the cost is I think it's very expensive. I think 300 million until to $1 billion US, but it might be in Indonesia still similar but different currency, maybe in rupiah. Yeah. Actually, the idea I think is very important, but I think in Indonesia sometimes it's still individual idea. I think we need in the future. I think idea must become to the idea institution, not individual. So after we get the training target and then we get the lead structure, I think actually we can directly to create clinical trial with this compound is possible very active, or directly can be preclinical and then to continue to clinical. But if not so active or difficult to synthesis or to isolate, I think you need optimized lead structure for to get the easy drugs. I can read the yeah, actually in the drugs come from the first from natural product, yeah, but actually because there are many problems in the natural product, I think some industry developed to make synthesis, derivative, and also full synthetics. This one is almost the drug is mostly from the synthetic one, from straight synthetic, yeah, because any problem of the to get the lead compound. Actually, in general, actually the natural product remain an important some source of new structure. But actually, for this, I'm more detailed. This is the compound is for anti-cancer, for synthesis 25. The major is for anti-cancer 25 drugs, and for antibacterial 19. And totally, actually, the almost drug is from synthetic. From the source, I think the source of natural product is from plant, bacteria, fungi, and still limited in marine mappings because the fund is also not easy to get the sample or maybe to get to scale up the industry. Yeah, so this one is step for discovery and development from natural product. I can national screening from can use plant, leaf, bark, and microbial from marine, amino, and so on. And then you must isolate the purification and determination structure. And then after you get the structure of the compound, you can also optimize identification the pharmacophore, and then you can optimization the compound with use derivative or analogs to get active pharmaceutical ingredient. Yeah, so I just before to this one, I inform you. This time in 1960 to 1980, conducted a study research for anti-cancer from 2,000 plants, but it was unsuccessful. Then continue in 35 plants, particularly from library extract, and succeeded in finding the anti-cancer drug Taxol and condition. Actually, for Taxol and commercial, I think this time is need around 60 years for under commercial drug. Also, the problem is I said the integration isolation as a compound patient is very small amount. It's also the problem. And then, for example, Taxol only 0.01% and very slow growth of the U3. For structure definition, sometimes it's difficult because the molecule is big or complex. Then structure of this complex, therefore difficult to synthesis and intervene the pharmacophore. It takes much more effort to mix a lead compound into drugs candidate for solution selected major compound in the medicinal plant as precursor, like for example, Taxol is synthesis with semi-synthesis from Pakatin tree from Taxus pacatana, and then synthesis and 4 step you get the Taxol. Yeah, so just in general procedure how to screening compound from some extract or from plant. Actually, is the morning just you heard from the Professor Koichi Batasi. And then actually the one is for screening is possible. You can get the active compound is extract more less than 100 ppm, but it's possible. Most better is most active is you can get the activity less than ppm. For example, this one is from Cytoxylum aromatica from extract. When this compound is very active because the IC50 2.9 microgram per milliliter. But also we need to also assay for cytotoxic. If the 50 more than 100, I think the extract is very toxic to the normal cell. This is the general. Was the unit fractionation to separation the non-polar fraction, semi-polar fraction, and also polar fraction. And then we create screening again. If you have the most active, the best activity, we continue to the fractionation. Also, this one is probably you must know how to the purification the compound, because sometimes if you don't know how to correlation the L1 from CRC and confirmatory, I think you can take time and lose chemicals. You must know the best condition how to the purification some fraction to column chromatography, or if possible can also use SPST. From all this one, I think this traditional screening, but you must step by step to screening from extract fraction until get the pure compound. This one actually 2573 as this IC50 is I think very strong. And the IC50 is more than 160 atoms. This compound is selective index or 69 indicates that the compound is very selective and very good for to develop content. Yes, one. Yeah, this one actually, this component is still very complex. I think this time it also takes time how to the definition the structure. But actually, this component actually this project is a started project with the University of Indonesia and at this time is Kobe University. Because this component actually the potential to develop the extract or compound actually potential to develop for anti-HCV. Yeah, so the other one is just method for how to fast screening. You can use the TRC photography and then actually you can use from the fraction active fraction active subtraction, and then you can TRC and then you can test for antibacterial, and then you can, if your compound active, you can see the clear zone in the TRC spot in the antibacterial assay. And first of all, sometimes the problem is I said the compound is very limited. This time this compound I only get around 1 milligram. I think this one is our PhD student, but the NMR actually one was instrument is important one because it's compared with the SMS, because the NMR you can know how to the stereochemistry, like double bond is cis or strand, of the position hydrogen is ortho or meta and so on. So you must know I think you must for chemistry, I think you must know how to reach the NMR spectrum. Actually, sometimes it takes time, but also I think to get the spectrum also in Indonesia takes time, because maybe in Indonesia only one this time only one, but now is have 5 NMR in Indonesia. Maybe I have friends from the Karachi or from Pakistan. One campus have 12 NMR. One campus. Maybe in Japan, one campus, every campus have 5 NMR in the lab. So it's comparing this. So it's funny something to take time in, and then if you have this area, this one is aliphatic area, it's double bond area, and then this one is aromatic. You can combine this functional group. You can predict the structure. This one also have good activity for antibacterial. After this one is in your compound, but with simple structure. Also this one we started from this method, the autography method. If you have background the chemistry, you can use this one as the glucose function. This one is double bond and then also double bond, but it's very downfield indicated the compound is contain nitrogen atom. So basically we can predict the structure and then compare with some literature. Yeah, I think this time only we can got the one only got the proton MR, because the sample very less than 1 milligram. Also this one our research just to give the, because when I student my professor said only one in few minutes he can determine the structure. So I think the spectrum I think not complex, like simple structure, because they have many methyl singlet, 6 methyl, but 2 is separate. So in this case, this compound is like this. The frame is like the percent type derivative, and then it's double bond. Contains double bond one in here, and then 2 carboxylic acid in 100 XT ppm, and then this double bond. I guess also this one is in CS2 and CS. Yes, so this one we can predict this structure. This one also we support with MS also when to the NMR. This one is new compound, but is compound, but yes, simple compound to this type. Actually, I think many methods of auto screening, faster screening, screening, but it's actually in general you need more vaccination for the use. Normally you can use methanol and then extract and reset, and then you can also use good extract from library if any get active extract. You can continue to SMS, and then after you can application to get the more. And then if you get the, for example, this blue one is active, you can then replication and to get the more enough sample to measurement like NMR or MS, LCMS, MS. Yes, I think I will be able to ask when the basic field, why the is field, because the two major reasons, some major reasons. One is the pharmacokinetic, because 39% field with the deficient in absorption, diffusion, metabolism, and elimination, and 10% field due to the lack of efficacy. The third is toxicity, and also actually the last one is maybe for commercial and miscellaneous problem also. This one, actually for this one we can use this rule of 5 to cut off the value for each this parameter. For example, if the rule states that pure absorption or formation is more like when the compound have too many hydrogen donor or too many hydrogen bond acceptor, and the molecular weight is too bigger, more 500, and the log P is I think is the parameter for polarity. If the log P is more than 5, I think the compound is very non-polar. So this rule is by many as useful guide in the design before computation or synthesis. Actually, the optimum condition for repeat schedule of 5 is based on data. This diagram from to C log P is polarity and the molecular weight. The ideal compound is optimum condition is if you have molecular weight is around less 500. The log P is around optimum is 3, around 3. You can this log P can calculate with some software. Yeah, I will just so why is lipophysics is important to drug development, because lipophysics is a key physical property for predicting biological activity of drug. The body is made up fatty compartment of cell membrane and aqueous compartment, inter and intracellular fluids. Drug need to pass between different compartments to get to their site of action. A suitable balance between lipophysics and hydrophobicity is essential for to drug to have right transport potency be equilibrium. Okay, you know, just one of many drug for antiviral, like for example, oseltamivir, molnupiravir, is one of have. I think this because some lipophilic functional group like ventilator, and accelerator, and also this one also we have the functional group of ester to increase the lipophilicity. That's it. Just for example, just when I just finished the PhD in 1906, I think this compound is very popular because the University have the project is molecular national. This one is yes, they have many cooperation with some, and then development curcumin for anti-cancer. Actually, the curcumin is so activity is good activity, but if you make some derivative like pentagamma this compound and pentagamma 1 is this structure, the activity will increase to time until 10 time compared with the curcumin. But you can check the log here. I just checked on the log here. I think this one is a rule of the Lipinski. So also structure application for lead compound development for every drug, not cancer only, but you can from other drug. Then I think you can use from existing structure material because and then can design some simple derivative. Use expensive reagents, screened by Lipinski rule, screened by docking with some receptor, for example for anti-cancer, anti-malaria, and so on. I think you need to develop the quick easy. I think that from Japan also already development because some country also develop also on the development, let's say in not in micro PPM but also in nano say. Yeah, actually this compound is a compound we can cut from the some microbial like this one can isolate it from 760 SP, this one lopatacin is commercial drug. You can also use for such material, and then this one this time isolated this penalty carboxylate. Also we can the analog synthesis, and also this one sulfochlorine is good for anti-diabetes and from isolated from aspartic. So the all methoxylamide is a major compound from Athena galanga, from oil Athena galanga, and this one is citronella oil. I think it's a cheap one. Such material you can get easy citronella or citronella. Also, I think in Indonesia have produced quinine. So there is also potential to develop for drug. Yeah, so this compound is the same. I isolated the major compound, but the activity is not strong. So how the structure modification to this compound? I think there's many methods for chemists. How the first is change substituent, extend the structure, change the ring. There's also the ring variation. You can change the isotherm maybe to O to G to N to R to sulfur, and this one I think is also important to simplify structure. If you have a complex structure like taxol or like you can make a simple compound. I think in this morning you heard from the it's forced to optimize the drug is you can also get combination drugs, but you can also how to repeat two drugs or two compound become one compound to get more active compound. After the all method to how to increase the intensity, decrease and water solubility, and so on. This one is I think many methods to hold on the optimization the lead compound. Yeah, the one method is actually this very simple one. I think you can use from the start from the simple one. Yeah, so if you have the alcohol or amine, you can acidification or acidification. As amine, you can get some acid like aliphatic or aromatic acid. And then this method, I think this general method. Also, you can carbonyl group can reduction with and then is the ester can change to carboxylic acid or directly hydrogenation to get the strong reduction like lithium aluminum hydride to get the alcohol derivative. That's just a story from the whole aspirin. I think this is long time ago, yeah, because in 1660 it was done get the drug from the salicylic alba. This one is salicin, but this time is any side effect because and then in around 100 years in 1x you need a hydration and oxidation to get salicylic acid. It was more effective, but still not bitter, but still a side effect was gastric bleeding. Also, in there Then the buyer make acetylation from this acetylation, and we get the acetylation is less irritating as to active drug. Also, this compound also was repositioning the drug from analgesic to become the blood thinning properties or antiplatelet. But now also this compound aspirin also used for development for anti-cancer. And still, for example, maybe you know this artemisinin is actually drug for anti-malaria. I think you know in 2015, Professor Yu from Chinese get Nobel Prize. This was focused on the for the contribution to discovery of artemisinin. But now artemisinin also you can use for anti-cancer. Yeah, the one is because artemisinin is sometimes is resistant to anti-malaria because so some you need to develop some derivative. So the heterogenation actually is substitution because it's any ether group, the ketal group. And then you need heterogenation, then methylation or acetylation to get methyl artemethyl as ether. Then we also can do this one for actually the one is more better activity is derivative. But maybe the problem is for heterogenation. I think this one is an isomer. Maybe you know. Also on this asymmetric synthesis, I think we need to speak catalyst for asymmetric synthesis. Yes, and just I think just proposing some artemisinin for anti-tumor actually many application for like artemisinin or I think so. Also compound is two compound anti-malaria like. This one is quinine, and then this one is artemisinin. Also combine the structure. Also, the first unit oxidation is one to oxidate, but the first for oxidation you need to processing group by PPDMS correct. Yes, processing group because if you don't protect this one, it's also become carbonyl. So we need processing group first in the oxidation group, and then oxidation with joint reagent. And then can combine this quinic acid derivative with artemisinol, become this compound, and then you can process them very easy with tetrafilament to get the use result from some. The HIFIS is showing the more strong activity compared with the quinine, artemisinin, I think there 15 times. But the mixture also so activity, the quinine and the also have activity, but compared with the compound is less. The HIP compound is more better time compared with the mixture. Yeah, depend on you. I can if compared in pharmacology can for easy. I think we can mix mixture, but this one more better activity can combine. Prohibit the two compound or two drug for become one drug. And so I think you can some information for simple from citronella. You can amination with filamin and then got this one is actually very strong for anti-cancer, but this one not selective. I think this one is possible actually the selective index is for cell normal. This one is toxic. Our research structure is very important. Actually, it's very simple. We start from the compound ceramic acid from the citronella oil, from the galanga oil, and then we can use autocresol in it. In participant, we can cut the methyl from penylchroman on. But we get if you use solvent like chloroform or so on, with paratoluene sulfuric acid, this compound become ester. Then the activity is the simple one is so this one is log P polarity is very important, but this the activity for this cell line, the penicillamide is the best one for activity because the activity 0.5 compared with I think for starting martial acid around around 100 times. This one is compounded as a group, but the lactone group is not so activity, not increase. Yeah, increase but not sometimes decrease in this one. Yeah. This one the sample for photosynthesis and to say compound. Yeah, since this one I got many compound as well. This time I got around 3% from Garcinia in the rest, but activity is still low because the activity is more than 20 microgram per milliliter. Then we had recognition first, and then we esterification. The ester group with some we can use some many ester like with some long different chains, and then this one compound actually good for the activity become 20 times compared to the Calanon or because the activity more is strong activity with 1.3 microgram per milliliter also for this colon cancer and leukemia cell. And I think also our group make ester with the butyrate, and we can use also the 2-dimethyl butyrate. I think this one is like for ester group from the sympathetic. Then some the activity is the most active is from cysteine isobutyrate and cysteine 2-dimethyl butyrate compound, but this one is medium activity from the caprate. Maybe this one is too long the change. Yeah, this time also I started para-carboxylphenazine, which is also strong antibacterial, but also so activity to cell line leukemia cell in around 5 ppm. So I think this compound is minor compound from the we sorted from Pseudomonas coccinea from soil, and then we make some that's a simple compound, but not the you open the rings, and then we can more from aniline and salicylic acid, and salicylic acid with reflux with paratransferrin, we can got this salicyl analyte, and then so on. We can change this salicylic acid, we change with the hydroxychlorotic acid or chlorobenzoic acid or this compound. We can find the strong activity is this compound, the salicylic acid. Log P is good, and then binding we can docking also good for affinity, and then the activity is for this cancer is around 3 ppm, and from KB cell activity is 0.6. I think one is very one-step reaction, but we can cut the good activity. Yes, so we can easily get the sample within or synconidine from the peticil in Lempang. Can design some make ester or ether, some with some acid like valerial, chlorine, and so on, and then we predict with the binding energy and drug license properties of syncona with the glycoprotein HPGP. Also from the quinine or synconine also have good affinity to this glycoprotein. But the ester group also derivative also have also actually for example for quinine isopropanoate and synconidine isopropanoate and hexyl isonidine. And the other one also the other component is ether group, but not good activity. Then with synthesis, I think in more detail, the interaction with ligand receptor for SPGP, in more detail, I think. And then we synthesis the compound. So based on the quinine is also have activity around 11 micromole, and then for P47D is also good affinity. But for the other group, I think was the best one. This student, this my research is we got the very strong activity for this component. For compound 3 is quinine isoclarate. This one is very strong, but also this one is also stronger because the activity is less than 1 micromole per milliliter. This one is, I think I could continue this. This one, I think also I have the UK3 compound so activity for some cell cancer line, but for this one, some cancer line, most leukemia, most normal, and so on. Yes. And then we tried to how to develop this compound, because this compound, as this time I synthesis in 14 steps in 1 year. I think it's several years, and then but it takes time. But then we need to develop to open this chain as for more easy synthesis in 2 or 3 steps. The result is we can synthesize 3-hydroxychlorotic acid with glutamate. We can easy synthesis with glutamate and butanol, and then you can get this compound. After application, you can say this one is good. Energy binding is around minus, and then the acidity is become better compared with the UK3A. This one is especially for KB and YB1 is breast cancer. Also to check this position the hydroxy group in the ring pyridine. This one is position 3, one is second 2 C2. The activity still have good affinity because the activity for KB is still similar with the PDPGE, and the other one I think still just to even also still have the activity. Yeah, more simple one, I think one is you need two-step reaction, because you need to spend with butanol first for glutamate. You can directly to use the oxaloamine. Yeah, also the acidity is strong. And then we can resolve. We try again how to the thing to treat the N atom, because to come from 3-hydroxynicotinonic acid change to salicylic acid with oxaloamine. You can get salicylic oxamate and also have a good affinity, good activity for KP cell line with acidity 0. I think this one is not milligram. Yeah, I think it's microgram per milliliter. Yeah, it's wrong. This one I just got the certificate patent in this year, because patent initiative time to get the patent. And one of the candidate, the simple drug, salicylic, we also in vitro test, but the result is still, I think, still toxic with this compound. But the efficacy toxic is around 35 milligram per kilogram body weight. You can just show the cell tissue damage and cancer cell level is decreased. Yeah, it's one weight dose 30 milligram per kilogram. But my fish, this compound is still toxic. You need to more explore again to the other compound to in vivo test. Actually, you know, this simpacin is good candidate for the commercial drug for cancer, but the how to synthesize this compound, existing technology is to root hydrolysis and direct methylation. This one is lovastatin, and one is simvastatin. I tell you, simple. It's difficult to direct to methylation, but if you also you can the other one the method is one. Yeah. The lovastatin is the patent is unique for we finish this compound, we need to group first, and then speciation this compound, and then you can acidification this after hydrolysis, you can get the sympathetin, and then you keep protecting this group become the sympathetin. I think even like simple, but the reality is not easy to synthesize this compound. The time I tried to our student also not easy to this synthesize this compound, the sympathetin. So I think this idea, the idea is from the Calvi Pharma, and this time she support upon this research, and how to develop the product the lepasacin. But I am focused is our team is also develop produce lepasacin, but I focus on the how to make lepasacin derivative based on this computation, and then we synthesis the affinity code affinity, and then we can get this compound with the in vitro test for represent for total cholesterol or STL. This is the result from the interaction energy with mg quarter cluster. The hydrolytic acid is I think is the best one for affinity, and then we synthesis. The synthesis could yield around one step, but in 100 x%. And then we test in vivo we compare at the time I compare with sympathetin. I think this with the same concentration with the same dose 7.2 milligram. The actual for HCl is similar with compared with this also for the triglyceride and total cholesterol. Yeah, this is and then because this one is compare, we need to also compare again with the comparative cholesterol and with atoxacin is commercial drug. So but is in this data is indicated for the atoxacin one is with one dosis one is with the activity is I think compared with the atoxacin is similar with two doses. There's two is XTH, but similar with the atoxacin, but also for SCL, I think the DS2 is better compared with I think not so significant around. I think this is similar with the atoxacin for total cholesterol and HCl cholesterol. And the last one, I think I have this time I have just cooperation with Republic Indonesia. We also test some anti-HCV, but the lepasacin is not decrease but in 20 ppm the virus is growth 40 times in the 20 ppm, but also the other statin, but our lepasacin is also good candidate for anti-HCV because they can inhibit the virus growth. This one, the structure. The last one, I think we also this time our group also make some derivative from some compound from steroids like creatine, scopolamine, ditroglycerin, and tribromoskorin, and also the most active one is 3-bromoscorin with one, but it's also the selective index is also good there. But the good also have the best activity because and the less 0.1 ppm, and then selective index is very good there, more than 2000. And besides that, I think this one, the finished one is, I think, natural product is potential for development new drug, and isotope compound have been modified structure to optimize with change to substance and simplify and or repeat. This can be screening by using Schirol to control velocity and molecular docking with some receptor. To optimize seasonal compound, you can sequence reagent and step synthesis and simple method and suitable to scale up. The last one is some derivative analog, I think, have potential to develop a cancer drug candidate or also for anti-cancer and anti-HCV. Last one, I see to experiment to some Ministry of Science and Technology. This time I got some funding from RIPI, also from GSP, Lab, also in cooperation from Osaka University and Kobe University and California, back to USA. Also this time I with JS Aero, some industry unit collaboration with KLBS, Texas America, Kalpi Pharma, and some small industry, Indonesia and Synchrona, and we have also some cooperation with some university, and also our project is support also some our research is also support by some many PhD and master and some undergraduate. So thank you very much from our team. And also thank you for Hitachi Global Permission and Mason Step from HPP Center, and also our researcher and participant for joining our this seminar. Thank you very much for your attention.
Thank you very much, Professor Hanafi. All right, we will move to the question and answer session. I see one of the question in the chat box from Bapak Agus to Prof. Hanafi. From the research result in the Aromatica, you found so many fractions, around 160 fraction. That what is the among fraction which can kill the COVID-19, and they are synergized, and how about the interaction among the fraction?
Okay, thank you for your question. I think you asked about how to this slide. I think
yes, I think this slide, Professor.
Yeah, I think actually I think the material component also come from the semipolar fraction. So it's from the compound we can many fraction we can get after different recombination, but we need also select it again. Selection again with so again this one is more better because check one by one from this say. Yeah, I think the proxy one, the fraction from I think the active one is from fraction 95. 95 is the best one, so we can continue this compound, and then again, not all compound we isolated, but only have the best activity, we can continue to get the structure. Yes,
so you
answer?
So you screening all the fractions selectively, Professor?
Yeah, of course we must screening selection. This one give the activity, good activity, the best activity.
So
the step by step you need verification which one the active, you can continue to the verification again by SPLC or so on.
Continuing the question, Prof. Hanafi, what is the result for the eucalyptus herbal research, the minyak kayu putih related COVID-19, the collaboration from BRIN or the IPB? What is the marker?
I don't not yet as the eucalyptus or minyak kayu putih, not yet to. I think some publication have the potential for COVID-19, but which one? I thought the major one is I think I don't remember I think the major one is I think is good potential we can develop for new derivative for to make good activity. yeah, so we not get to test the activity for COVID-19 because we don't have facility for the COVID-19. Also for this let's say antiviral SCP, we join with the university in Indonesia because they have the facility PSL2, yeah. So this component, no, yeah. This time PP not yet have the PSL2.
Going on with the anti-cancer potential of the herbal medicine, what is the marker drugs that potential for the anti-cancer with the high effective as a candidate for the medicine?
Yeah, actually many plants you can select, but for because we need to how to based on the I think many drugs have the repurposing like for the for like artemisinin you can easy isolation. In China you can cut more than 2% from the plant, but Indonesia have only low yield to isolation the synchronin. But this compound also potential for anti-cancer, yeah. And the cheap one, I think you can, because I will start from the one compound, yeah, for cheap compound, like quinine. I think quinine is a product from the peticil in Lembang, so we can make some derivative, and then we can put some derivative. Yeah. I think this. Yeah, I started from the last one. Yeah, I think. Yeah, I think this one is a potential drug. I think yeah, photosynthesis is not so difficult. I think there's a simple only one step, and with good yield, I think there's potential to continue to the in vivo test, not yet to in vivo test.
So I'm a little bit curious, Professor. Meanwhile, I'm waiting for another question in the chat box. I would like to ask. So, except from the difficulties to isolate the major compound from the Indonesian's related plants or medicinal plants, what is another difficulties for us to develop the new candidate drugs, especially after the modifications of these drugs within by utilizing the simple synthesis?
Okay, you mean is also the problem, but among from natural drug sometimes is the structure is very complex, and then the yield are very limited. Yeah, so actually we get depend on the drug, yeah, like artemisinin or like fluvastatin actually it's already commercial, but can use also directly to the commercial drug to develop some derivative. But for isolation, yeah, depend on the structure. I think yeah, depend on what kind of structure, like this compound, like yeah, I think not easy, yeah, to synthesis this one, but sometimes like what's compound this bonding synthesis. So the compound is like this one, separantin. Yeah, separantin is I think is B compound, yeah, B like similar compound to compound, yeah, like lipid compound. But also this one is oligomer is also the like some like trimer oligo. Yeah. But for this one was difficult to say. Can develop also isolate the precursor, the other precursor. Maybe you can isolate the other compound like the monomer this compound, and can optimization again like also like similar like the taxol. Yeah, taxol. I think the first is very difficult to this time, yeah, because the only got the 0.051%. So for chemists how to isolate this compound with this time, we can put this bakatin tree from the bacata, and then we synthesize it to run for 4-step reaction. I think this one is also we can commercial like semi-synthesis. Yeah. To answer your question.
Thank you very much, Prof. Hanafi. One last question, Prof. Hanafi. Very brief answer, please. The question is from Bapak Asep Arifin. Many thanks to Prof. Hanafi. Quinine derivatives, the number in your slide is number 3, has a high potency for the anti-cancer drug. Are you plan to doing the next research, especially for the in vivo study or clinical study? And more specific question is when?
Yeah, first, I must team again, yeah, because I think it's not individual project. I said must coordination can by our team, and it's possible I will to make first to scale up for synthesis. I think we can collaboration with PTC. Yeah, I hope so. Is we can to continue this one. I think was the one compound is the best one. The compound I got the activity. Yeah. The other one is I think around 1 ppm. This one I think very strong. Yeah, I have any. We can get because that is the problem in Indonesia. Sometimes one is like individual research. Yeah, sometimes everybody don't care what is. Yeah, I think I hope we can continue next year, or because until now the host program host is in from orchestra, not yet. Yeah, I think we need. I think somebody, but for young scientists, I think many young scientists you can continue if you interested for this component.
All right,
Yeah,
thank you very much. Yes.
Yeah, I hope so. We can continue for. I think many lead compound to continue to in people.
All right, thank you very much, Prof. Hanafi. Finally, now we reach the end of the presentations. Thank you very much, Professor Hanafi. I would like to return the screen to our MC, Dr. Yudi. Are you here in the screen? Okay.
Thank you very much.
Yes, thank you very much, Dr. Yudi.
Okay, thank you, Dr. Zetri. I would also like to thank all the speakers for the insightful presentation, and to the audience of this webinar for their participation. This event has been performed successfully, and finally allow me to close this event. Thank you very much for your kind attention.
Thank you.
Thank you very much.
Thank you.
Thank you very much, Hanafi.
Terima kasih Prof. Hanafi.
Terima kasih banyak, Prof. Terima kasih, Prof.
Mas Yudi.
Terima kasih, Prof. Yuli.
Sukses ya.